Now, one of the questions people have is, how can you say that your NK cells can cross the blood-brain barrier? There are some natural killer cell products that are out there in clinical trials where they have to directly inject their NK cells into the brain tumor bed because they don't seem to cross the blood-brain barrier. Well, this is a study that showed the role of a chemokine receptor called CXCR3. And what that is when they looked at patients with really chronic brain issues, whether it be multiple sclerosis, Alzheimer's disease, Parkinson's disease, they found that those areas of the brain that were affected had very high levels of CXCR3. And more importantly, when they looked at the T cells in those patients, they also had very high CXCR3 expression. So the feeling is, particularly in Alzheimer's patients, that they found CXCL10 positive astrocytes, which were frequently associated with amyloid deposits. So you have amyloid deposits in those areas that have high CXCL10 expression. The T cells have very high CXCR3 expression that can cross the blood-brain barrier. And in fact, when they did autopsy data, where they looked at patients with chronic Alzheimer's disease and autopsies and stained the brains, they found that those specific areas, the neocortex, the hippocampus, the striatum, and the cerebellum, had very, very high expression of CXCR3. And that's in fact where all the proteins seem to accumulate and where you lose activity on a PET scan. And the idea is that you had very high protein deposition that led to very high CXCR3 expression. And then the T cells also had the CXCR3. So they crossed the blood-brain barrier, went specifically to those areas, and started to cause damage and inflammation due to the protein accumulation. And the same thing that you'll see in Parkinson's disease is this very high amount of CXCL10, CXCL11, and the CXCR3 that I just mentioned. Of note is when we activate and culture our cells, you can see from day 0 to day 17 how we get a very, very high CXCR3 expression. And when we've done studies looking at cell migration, our ability of our NK cells to go where the action is, you can see that when we look for CXCL9, CXCL10, CXCL11, that in fact our NK cells had high migration ability to go to those areas. So one of the ways that we would say that we feel our NK cells can cross the blood-brain barrier is specifically through this CXCR3 expression, that they're basically following the T cells to where the action is, and then going after the T cells based on the mechanisms that we discussed on the previous slides. So when we look at how our NK cells work, we think our NK cells first cross the blood-brain barrier versus CXCR3, and then through the presence of DNAM1 and NKG2D, they can identify and eliminate autoreactive T cells, while at the same time helping to secrete interferon gamma to activate macrophages and microglia to then help to eliminate the alpha-synuclein or amyloid proteins. The final thing that they can do is also through NKG2D identify and eliminate some damaged neurons. So it's a much more comprehensive way that NK cells function in the brain compared to just removing a protein. So we wanted to test this out on some patients. And while most of the clinical trials focus on mild cognitive impairment, we really wanted to take patients with really end-stage disease to see if we could make some improvement and that there would be no doubt as to why the patients got better. So the first patient we came across was a 38-year-old when we met him, but at the time he was diagnosed at the age of 34 with advanced Alzheimer's. He was a Stanford a grad who was working in the Bay Area as a programmer. He kept getting lost on his way to work. Eventually was found to have a PSEN1 mutation at UCSF, which basically guaranteed before the age of 50, he would have full-blown Alzheimer's disease. When we met him, he was already in hospice requiring 24-hour care. You can see his PET scan that showed striking decreased activity, which was consistent with Alzheimer's disease. We actually reached out to Dr. Ming Guo, who's the head of Alzheimer's and Parkinson's at UCLA. Might she be able to see this patient before we treated him? She saw him and said this was probably one of the worst cases that she had seen. When we met him, he could not talk. he could not get out of a wheelchair and he could not feed himself. So after obtaining informed consent and discussing this with the family, who agreed to allow us to treat him, we proceeded to give him several doses of his own enhanced cells. Now, remember, when we met him, he could not walk, talk, or get out of a wheelchair.
All right, Dan, let's go. You have to unbuckle your seatbelt. All right, Daniel. Don't forget, you gotta what? What do you gotta do? There you go. Nice.
So not only could he now get out of the car, which I would suggest is more difficult than getting out of a wheelchair, he was then able to comprehend that he needed to close the door. Here's him eating.
Daniel, how's the food? Good. What are you eating? You like it? Yeah. Awesome. You want to try eating with yourself? Yeah. Okay. Do you remember how to use a chopstick? Yeah. Okay. Well, let's see.
This is him. The nurse took a picture of him in his nursing home while he was sleeping. And you can see his leg was trembling as if things were reconnecting and reawakening. So again, here's one example of a patient that was very, very end stage, was really not given any other options, and who has had a dramatic response.
Daniel, how's the food? All right, Daniel, let's go. Good. What are you eating?
Next is a patient of the head of capital markets at one of the leading banks, who I met at a conference several years ago. He reached out to me and said, you know, my mom has advanced Alzheimer's disease. Do you know anything or anyone that's doing anything innovative? We mentioned to the family that we had something that was not yet proven, but that we would consider making it available for her. We had started her treatment pre-COVID in a country that considers a non-genetically modified autologous treatment as a treatment, not a drug, and does not require FDA approval. But then COVID hit, there was a 2-year lapse, and we were unable to treat her again until last week, where the US FDA granted us approval to bring her treatment to the United States and resume treatment under compassionate use. What's really interesting is when we first met her, she didn't remember her kids' names, her husband's name, that she lived in New York, or any of those things. But here her husband, who has given me permission to share this video, talks about the experience.
Can you describe really what happened, her first experience with this?
The first six treatments?
Yeah.
We saw a significant benefit in her cognitive abilities. On a personal test, it went from a score of 8 to 32. And then we did 2 other tests after those visits. The score remained high for 6 months, but was declining and continued to decline when we stopped. We then did some testing before this treatment today, and we saw a score of 6 or 7, and we're looking forward to testing.
So here you have somebody describing his wife's experience before. And again, we just got approval from the US FDA to resume her treatments after a 2-year hiatus. And we're very anxious to see if we can get her improving again. And that will be the first question. But this is part of our larger goal to get a full USA approved IND and begin clinical trials in the United States in the next few months.
Can you just...
This is another patient that was treated in Japan, who's from Korea. Japan is another one of those countries that views this as a treatment, not a drug. And you can see she went from a scale of 12, which is severe dementia, to a score of 22, which is mild dementia. We have also treated somebody with advanced Parkinson's disease who has had marked improvement as well. Dr. Guo at UCLA saw this patient pre and post treatment and did also confirm that this patient had dramatic improvement. We have a current trial underway for advanced Alzheimer's disease. Unlike most of the trials are looking at mild cognitive impairment and trying to slow progression, we're trying to take really advanced Alzheimer's disease and show real disease modifying improvements. We have signed an agreement with the Parkinson's Foundation, and what they're really helping us to work on accelerating the path to get approval to do a Parkinson's trial in the United States.